Welcome all students in this new semester. In this paper of nanobiotechnology, we will learn about this interdisciplinary subject. This paper is nanobiotechnology. Subject code is BTL0717, and this paper, as it is clear from the name, it has two parts: nanotechnology and biotechnology. I'm sure that you may know the potential of biotechnology that you are studying. Biotechnology is a science which deals with study and modification of living organisms and also biomolecules to enable cost-effective manufacture of various products and vectors. Biotechnology involved expertise in variety of fields from biology, biochemistry, molecular biology, molecular engineering, genetics. And for this, we have to work for development of many devices. The development of many devices fall in the field of nanotechnology, because all the living organisms based on nanotechnology, based on nanosystems, on small systems. We will study many things about nanotechnology in this paper. This paper consists of few units on nanotechnology. How can you develop materials, synthesis, and then we will move to biotechnology, nanobiotechnology, and how the nanotechnology help to create biotechnology. So lot of things, lot of excitement you must have with this unit, with this paper, because it has huge potential to explore your field. We will discuss those issues, those points, and of course this will also help you to develop your overall concept in the experimental part of biomaterials. The lecture will start with learning outcome. What you will learn with this lecture. We have also introduced here subject student respective learning outcome. Then we will move to nanotechnology, basics of nanotechnology, biotechnology, nanobiotechnology. We will get the brief of these. Then what is the significance of nanotechnology? A brief history, who have started this and what is the present status, and finally the summary and conclusion. The whole video or presentation is prepared to keep you alive, to keep you attentive in this field, where you have to work hard, you have to learn to develop new devices, develop new systems. It's a beginning, and we will learn about significance of nanotechnology with why it is so important to learn and about it for biotechnology students. We will open a new way to think, to explore in this talk. We have started a process of writing these words SELO, subject effective learning outcome. These are some effective outcome. We have developed a few learning outcomes, and specifically we are entering here to give you the flavor of this. At the end of this, it will add something, ability to solve problems through application of theories, the way we have developed the theories. And such type of lectures also help in innovative thinking skills, critical thinking skills, and of course you will get the knowledge of how to collaborate with others. Whenever we use such terms, nanobiotechnology or bio-nanotechnology or nanobiology, these terms are refers to intersection of nanotechnology and biology. Yes, these are two terms. One is nano and one is bio. The subject is one that has only emerged very recently. The newest subject we are having. And these are the subjects or that serve as blanket term for many other technologies. Before moving to nanobiotechnology, we must realize that any science, any technology is for welfare of human being. Either we develop some devices for the human being, or we develop many organs, many systems for the welfare of human beings in life sciences. Ultimately, we are trying to search basics of life sciences. So biotechnology is something to improve the life expectancy, to improve the quality of life. They are doing a lot, but for development part they need a technology, and many part of technology came in this range, nanotechnology. So this is one of the newest and one of the highest potential branch. We can see through this that there are two things. One is structural engineering and there is genetic engineering. Structural engineering talk about many structures at a small level, and this structural engineering help a lot to develop new materials. And development part come in the range of nanotechnology. From here, the genetic engineering helps you to develop biotechnology. It's a part of that. And these nanotechnology and biotechnology, when they combine each other to develop new things for developing many collaborative effects like sensor monitoring, nanosensors. We need a lot of nanosensors. as we must know that our body or any plants all are full of lot of sensors, and we want to develop such sensors, then it's called smart monitoring nanosensors. The development of such sensors is possible only when we have a set of rules to prepare something, that is nanotechnology. It also helps in target delivery approaches for gene transfer, and also many applications like pathogen detection and so on. So that is something about this new field. We can conclude that this is a discipline in which nanotechnology are developed and applied to study biological phenomena. Suppose we have developed nanoparticles, and that can be worked as a probe or sensor or a carrier for many deliveries in cellular systems. So this type of work falls in the nanobiotechnology. Nanotechnology, as defined by size, is naturally very broad. This includes fields of sciences. The arena is as much wide diverse as it starts from surface science, then organic chemistry, molecular biology, semiconductor physics, energy storage, microfabrication, molecular engineering, and so on. The associated research and applications are equally diverse, ranging from extension of conventional devices, from physics to completely new approaches based on molecular self-assembly. Words are very fancy. Self-assembly. Yes, assembled by their own way, by their own forces, we will discuss here. This includes development of new materials with dimension on the nanoscale to direct control of matter on atomic scale. As we have discussed in the last slide, here are some examples. The scale that is shown here, 0.1 nanometer to 1 millimeter. And many things are shown here. You can talk about atom, which is about 0.1 nanometer. Then fullerene, about 1. Then DNA. Then if you move to further, then you move to 1 micron blood cells, or many other things. So whenever we are talking about nano, we are talking about bottom of the devices, bottom of the synthesis. What happened at the first atom or the first group of atoms? So that is important here. Whatever we saw, whatever we use, it has been started from the first atom. And this technology gives you the power, the wisdom, even to manipulate single atom. That is the best part of this technology. The history of nanotechnology actually traces the development of concept and experimental work falling under the broad category of nanotechnology. As we have discussed in the last slide, nanotechnology itself is not a subset. It's an umbrella. It's a canvas on which all the sciences may merge to develop. Sciences like physics, chemistry, biology, mathematics, computer science, all joined here. So whenever we talk about history of nanotechnology, we are talking about history of science, history of engineering and technology. Many things have been developed. Soon I will show you. On one side, we can say the history of nanotechnology starts from 1954 or 56. But we know the nano word, nanotechnology, is already being used in the ancient centuries. We will take some examples from also from there. But the important thing is, first, what is that and how this nanotechnology developed. Can you see the different colors bottle here? Yes, the colors are very attractive. And why? The colors are attractive, and I am saying the colors are of the same element. Surprising? Yes. The nanotechnology showed us even the same material of different size may give you different colors. Color is not the property of material. Color is the property associated with the size of the material. When we move below 100 nanometer, everything will change. We can analyze the things. These unique phenomena enable novel applications, which is not possible whenever we are working only with bulk materials. Amongst all technologies, recent technologies, nanotechnologies are amongst the great technological advance of recent time. Yes, it has huge potential to develop. It has wide spectrum to develop. It has huge potential in interdisciplinary research. Nanotechnology is not an industry, nor it is a single technology or a single field of research. We call nanotechnology consists of set of enabling technologies which are applicable to many traditional industries, which have huge potential to develop to cater many agency industrial implications. I suggest we must understand the emergence of nanotechnology. from where this word came, how we have developed, and what is the future of this. Here this is shown and how physics, chemistry, biology all merged here. In the physics, we started with electronics, electron technologies, then microelectronics, then material design, and after material design, we improved the designing of materials. Small size. All of you are aware of the small size electron devices. Then we started the searching of quantum effects and moved to electronic devices at nano level. On other side, we are studying a lot in biology, studying a lot in cell biology, molecular biology, and from there we started the searching functional design of molecules, designing of proteins, carbohydrates in the lab. On the third side, chemistry is also developing. We have worked a lot on complex chemistry and supramolecular chemistry. For development of a device, chemistry helps a lot. That is why they merged together. Biology entered in this region to improve the sustainability of the devices, flexibility in the system. And that is why now our approach is integrated. We are exploiting many principles, many laws, many properties in an integrated way. Although nanotechnology is a relatively recent development in scientific research, the development of its central concept happened over a long period of time. It does not begin here in few days or few months. It took a long time to develop. However, we can say it has started from something in the second half of the 20th century, and then the interest increased in the last 20 years and from 1980. But we have many examples from the ancient past on this. There is plenty of room at the bottom. An invitation to enter a new field of physics. This was the title of one of the most celebrity lecture by physicist Richard Feynman. This lecture was given by Feynman in December 29, 1959. Yes, the evening of December 29, Caltech University witnessed one of the greatest lectures of this era. There is plenty of room at the bottom. In this lecture, Feynman considered the possibility of direct manipulation of individual atom as a more powerful form of synthetic chemistry than those used at the time. Feynman was the person who thought about manipulation of atoms, manipulation of matter at atomic scale. He was particularly interested in possibilities of denser computer circuitry, microscopes that could see much smaller than the possible. And you can imagine how visionary this person was, that these things were developed after 1950. In this 59 lecture, he suggested that it should be possible in principle to make nanoscale machines that arrange the atom the way we want and do chemical synthesis by mechanical manipulations. That is very interesting thing. He talked about manipulation of atoms and the microscopy in this lecture. And this dream came true after 1980. Although the version of the talk were reprinted in a few popular magazines, it went largely unnoticed and didn't inspire the conceptual bringing of the theory. Now, beginning in the 1980s, nanotechnology advocates cited it to establish the scientific credibility of their work. You must realize this. Up to 1980, nobody will give much credit to this lecture. Thank you. In the next lecture, we will discuss various properties.